The overall goal of this procedure is to visualize patterns of mRNA expression in developing fetal mouse lower urogenital tract tissues. This is accomplished by first synthesizing a digoxygenin-labeled riboprobe from a PCR-generated template. The second step of the procedure is to generate thick-cut sections using a vibrating microtome. The third step is to conduct an in situ hybridization in lower urogenital tract sections contained in customized baskets. The mRNAs are visualized through immunohistochemical detection of bound riboprobes using BM-purple precipitating alkaline phosphatase substrate. Ultimately, microscopy can show the spatial and temporal localization of low and high abundance mRNA transcripts in the fetal mouse lower urogenital tract.
The main advantage of this technique over existing methods, like immunohistochemistry, is that riboprobes are easy to generate and can be made to recognize most mRNAs in the entire mouse genome.
Though this method provides insight into mRNA expression patterns during fetal mouse genitourinary development, it can also be applied to other organ systems, species, and developmental stages.
Visual demonstration of this method is critical, as embedding the tissue in agar can be difficult to learn, and because slight variations in orientation can lead to inconsistent results.
At least one day before preparing the tissue, remove and discard the membrane from a 12 mm diameter millicell culture plate well insert. The remaining ring will function as a mold. Soak the rings overnight.